This topic covers the function and structure of the cytoskeleton, extracellular components of multicellular organisms, and cell connections of multicellular organisms. We will be covering microtubules, centrosomes and centrioles, cilia and flagella, actin filaments, and intermediate filaments. Under the topic of the cytoskeleton, crisscrossing the cytoplasm of eukaryotic cells is the cytoskeleton. Just like the name sounds, the cytoskeleton can give support and shape to the cell, like the human skeleton does to the human body. Unlike the human skeleton, the cytoskeleton can also be involved in changing the shape of the cell, transporting organelles and molecules within the cells, and so on. The cytoskeleton is composed of three types of fibers: microtubules, actin filaments, and intermediate filaments. Microtubules are the largest of the fibers. They form the spindle that separates chromosomes during cell division. They form the internal structure of flagella and cilia, and they help maintain the shape of cells that don't have a cell wall. They are also involved with transport within the cell. So a vesicle is attached to a cytoskeleton by a motor protein, and this motor protein walks the vesicle along the microtubule. It's kind of like a highway for the cell. In animal cells, microtubules radiate out from a region near the nucleus called the centrosome. It is thought of as a microtubule organizing center. Within the centrosome is a pair of centrioles. The centrioles are composed of 9 sets of bundles of microtubules, 3 microtubules per bundle. The centrioles function in cell division. When the cell is ready to divide, it makes a copy of all its DNA so that both cells can have a copy. The centrioles move to opposite sides or poles of the cell, and a bunch of microtubules radiate out from the centrioles. We call this the mitotic spindle. The mitotic spindle makes sure one copy of the DNA ends up in each new cell, rather than 2 copies in one cell and none in the other. Eukaryotes have structures for motility, or moving the cell through the environment. In multicellular organisms, one of these structures, cilia, have been adapted to moving materials in the environment past the cell and its tissue, but in this class we will be focusing on unicellular organisms. Flagella in eukaryotes have a very different structure than prokaryotic flagella. Eukaryotic flagella are composed of bundles of microtubules in a 9+2 arrangement, like in this micrograph. 9 bundles, 1, 2, 3, 4, 5, 6, 7, 8, 9 bundles of 2 microtubules each are arranged in a circle around a core. ATP is used to pull the bundles one way or the other. The result is a back and forth motion or a rowing motion. The flagella is attached to a basal body within the cytoplasm. The structure of the basal body resembles the structure of the centrioles. Cilia are composed of the same 9+2 arrangement of microtubules. They are basically short flagella, and organisms that have cilia usually have a lot. Actin filaments are the smallest of the cytoskeleton fibers. They are found in highest concentration under the cytoplasmic membrane. They are involved in cell division, contraction of the cell, cell extensions, and pinching off of vacuoles and lysosomes. Intermediate filaments are the medium-sized fibers. Their main role is in structural reinforcement of the cell and of organelles, such as the nucleus. In multicellular organisms, they are involved in cell-to-cell connections. Under the heading of extracellular components and connections, we will be talking about the cell walls of plants, the extracellular matrix or ECM of animals, and intracellular junctions. Plant cells have a cell wall that protects the cell and helps the plant have rigidity, sort of how a human skeleton provides rigidity. The cell wall of plants is composed of cellulose, the most common polysaccharide on the planet. Animal cells do not have cell walls. Instead, they have what is called the extracellular matrix, or ECM. The ECM protects the cells by cushioning them, keeping them moist, and acting as a barrier to pathogens. The EMC also connects animal cells to each other, helps with communication between cells, and helps with cell movement within developing embryos. The ECM is composed of proteins like collagen, polysaccharides like proteoglycans, and glycoproteins, which are combinations of carbohydrates and proteins, like fibronectin. Cells in multicellular organisms like plants and animals are connected to each other. Let's go over this diagram and talk about the functions of each of the cell connections. Tight junctions form very tight connections between animal cells. That's why they're called tight junctions. They help form barriers that don't allow fluids to leak between cells. For example, in the intestinal mucosa, tight junctions form between intestinal mucosa cells so that the contents of the intestine don't leak between those cells into the bloodstream. Desmosomes, or anchoring connections, are strong connections in animal cells which forms them into sheets. Intermediate filaments pass between the desmosomes of different cells, making them stronger. Gap junctions form little ports between cells. When they open up, fluid and small molecules pass directly from one cell to another. This helps with cell communication in tissues. Plant cells have junctions that are similar in function to a gap junction. It's called a plasmodesmata. They are membrane-lined pores that pierce the cell walls of neighboring plant cells, allowing cytosol and molecules to pass from cell to cell. This aids in communications of cells within plant tissues.